Hi, my name is Krina Patel, and I am an associate professor at MD Anderson Cancer Center in Houston, Texas, where I take care of patients with multiple myeloma and conduct research. And today, I will give you the update that we recently presented at ASH of the phase 2 registrational study of Anitocabtagene Autoleucel, or anito-cel, for the treatment of patients with relapsed and/or refractory myeloma. And this is the iMMagine-1 study that was updated. So this is the third BCMA CAR-T now that is attempting to get FDA approval. We have two, of course, already, both ide-cel and cilta-cel, that are approved in relapsed refractory myeloma as well as early relapse myeloma. And anito-cel is similar in that it goes after the same target, BCMA, but the way it goes after it is a little different. So here you can see that it's not a traditional scFv, but it's something called a D domain binder, which is sort of a small protein, which increases transduction of the CARs. So we think this is why our CAR cell products that come from anito-cel have higher CAR positive cells. And the other thing that it can do is come off, the off rate of this D domain from the target is a lot faster. And so again, toxicities may be lower because you can actually get the CAR-T off once it's done doing what it needs to do. So the original data from the phase 1 study for anito-cel showed a median PFS of 30.2 months in those patients that are fourth line plus. And so coming to our phase 2 study, again, iMMagine-1, these were patients who had prior PI, IMiD, and anti-CD38, underwent leukapheresis, bridging therapy, followed by lymphodepletion, and then anito-cel infusion. And overall, the patients that were enrolled, so far we've had 129 patients that were leukapheresed, and then 118 that received lymphodepletion, and 117 that were dosed. And again, you can see the reasons for discontinuation on the right here. And then the median follow-up for these patients for this study was 15.9 months. And here are the patient characteristics. And again, 100% of patients were refractory to the last line of therapy, 87% triple refractory, and 41% penta-refractory. Something unique here is that we had, 8% of patients that were able to get outpatient administration of their CAR-T. So the main thing that we've seen in response over time has been that the depth of response has improved as we present this data again and again and again. 96% overall response rate, but now we have a deeper response of stringent CR rates of 74%. And again, the time to stringent CR is 3.2 months. But the median time to best response for the patients has been 4.8 months. And then we also had more data on MRD, the minimal residual disease. So 10 to the minus fifth level, the overall MRD negativity was 95%. And for those patients that had an MRD undetectable at greater than 6 months, at least sustained for greater than 6 months, was 83%. And then looking at 10 to the minus 6 of 87 patients, 78% were still MRD undetectable. We're excited to hopefully have more data in the near future for the longer term MRD undetectability sustained at 12 months. And just to say that the last patient that got treated on this study was about a year ago. So then coming to PFS, again, the rates from 6 months to 12 months to 18 months have decreased over time. So our 18-month PFS right now is 67.4 months. However, predicted 24-month is 61.7 months. So again, the decrease, we hope that we have a potentially a plateauing happening as to why maybe just like the other CAR-Ts that we've seen, that now that's 24-month predicted right now is 61.7 months. And then safety-wise, we haven't had any, of course, CRS or ICANS since our last presentation, but we also have not seen any delayed non-ICANS neurotoxicities, including Parkinson's, cranial nerve palsies, or Guillain-Barré syndrome, and we have not had any immune effector cell-associated enterocolitis. We do see infections, but again, nothing that has been in the last since the last presentation in terms of grade 3-4 worsening. So in conclusion, anito-cel utilizes a novel synthetic compact and stable D domain binder where we think this increases transduction efficiency as well as a faster off rate that could help with potentially decreased toxicity. It shows deepening responses at a median follow-up of 15.9 months. The safety profile is predictable and manageable, and a few of our patients were able to get their CAR-Ts outpatient. So with that, we do have a phase 3 study that is ongoing as well, looking at standard of care versus anito-cel in the iMMagine-3 study that we're excited to hopefully have data for in the next year or two. So with that, just wanted to thank our patients, their families, all our colleagues from the different centers who are part of the study, as well as our staff and research coordinators and investigators. Thank you.